The most common question in everybody's mind is, where does the material for the exam stop? It never does. It just goes on and on. Kind of like me. So what I've decided to do is to stop the exam material at the end of this material that you see on the screen right here. Okay? So that's where the exam, that means I've got one more thing to talk about this morning, or this afternoon, whatever this is. And then that will stop the material for the exam. I have a review session planned for tomorrow evening, starting at 5:15. It's tentatively set for ALS 4001. And it's tentative because I didn't get confirmation back that I've got the room. I'm pretty sure that I do. So if that changes, I will send an email out to the class announcing the change. But I think it'll be in ALS 4001. So if you don't hear anything, it'll be in ALS 4001. Does that make sense? So if you don't hear something from me, it'll be in ALS 4001. Yes? 5:15 tomorrow night. I will be videotaping that as I've done before and making those available as quickly as I can. And I anticipate I should be able to get that uploaded sometime on Tuesday evening. That's my plan. Okay? Okay. So let's dig into the last of the material for this exam. And the last of this material relates to some other considerations of the regulation of cholesterol biosynthesis. So I told you, first of all, that HMG-CoA Reductase was allosterically regulated by feedback inhibition, meaning that cholesterol came back and bound to that enzyme and could turn the enzyme off or down, or however we want to say that. Okay? There are other ways in which the enzyme can be regulated. The enzyme can be regulated by phosphorylation. I'm not going to talk about that. Now, that's kind of complicated, and I'm just not going to talk about that. I may mention it briefly later, but not for this exam. Okay? The last way that it can be regulated, I want to mention here, is by whether or not the enzyme is actually synthesized. So that's what I'm going to talk about on the screen right here. So when we talk about regulation of enzymes and pathways, we've talked about different considerations for them. We've talked about allosteric regulation. We've talked about covalent modification. And now we're going to see a way in which that regulation is accomplished by either making or not making an enzyme. If you think about this, this is probably, all right, the most effective way of turning off an enzyme, because if you don't make it, there's no way you have to worry about regulating it. Whereas if you do make it, then you have other considerations. Well, the HMG-CoA Reductase's synthesis is controlled by what you see on the screen. All right? It's controlled by what you see on the screen. And what you see on the screen is looks a little complicated. And to be honest, the nomenclature here is rather complicated. So I'm going to try to simplify it for you. And make it more easy to understand, hopefully, as a consequence of that. All right. So we see, when we look at the ER, which is the endoplasmic reticulum, that there is a membrane protein that has three different segments to it. One called REG. This protein is called, the overall protein is called SREBP. And this is part of the confusion, all right? Because this is also called SREBP, this is called SREBP, and this is called SRE. All right? So, so I'm going to give some new names. We're not going to call it Larry or anything like that, okay? Although that might be even easier to remember. But we're going to try to adopt the nomenclature that they've got on here. All right? So, what are we going to do? So, we will call this complex right here, all right, SREBP-ER. Okay? SREBP-ER. This means that this is this protein. By the way, SRE stands for the steroid regulatory element. And you don't need to know that. And BP stands for binding protein. Well, as we can see, the binding protein is only a part of this overall thing. All right. So, this guy up here is SREBP-ER, right? SREBP-ER has all three components. All right? That's a component that includes the membrane part, right? That includes a part that is a regulatory part, and it includes a part that is a DNA binding part. We're going to refer to those as RMD. Many of you want to be MDs. There's an RMD up there for you. Okay? Now, as things are floating along, normal levels of cholesterol that are available in the cell, and the cell doesn't need to make cholesterol and so forth, this guy sits here as SREBP-ER. Okay? When cholesterol levels fall, this portion of the endoplasmic reticulum is shuttled off to the Golgi apparatus. And in the Golgi apparatus, there is a serine protease which is able to make a cut, as you can see right here. Now at this point, we're going to call this whole complex right here, we're going to call it SREBP-G for Golgi. So we've gone from SREBP-ER to SREBP-G. And SREBP-G has R, M, and D. Okay. Now, in the Golgi, when this cut occurs by this serine protease, the MD part of it gets separated from the R part of it. So SREBP-G has either RMD, or if it gets cut, it has the MD part. And basically, it's going to have only the MD part. The R part, what happens to it? Well, it sits there and does nothing. The MD part is the action part. So SREBP-G has, in this case, the MD. Well, we see that because it's been freed of this part over here, the R part, it can move in the membrane, and it moves over to another membrane protein called a metalloprotease. You remember metalloproteases were proteases that used metals to catalyze reactions. And this metalloprotease cuts the last part off. So what it's doing is it's cutting off the D from the MD. The D now is free to go to the nucleus. And in the nucleus, it binds to a DNA sequence called SRE. SRE is a DNA sequence, the signal recognition element, the steroid receptor element, SRE. All right? So the D part is binding to the SRE of the DNA. All right, so I'm going to step you through it, and then I'll tell you what happens finally at this last step. Okay? So we start out, we've got normal levels of cholesterol. The SREBP is in the ER, in the endoplasmic reticulum, SREBP-ER. When cholesterol levels fall, this complex is moved to the Golgi apparatus. In the Golgi, a serine protease clips off the MD away from the R, so the MD part can move in the membrane. It moves to a metalloprotease, which cuts the D part off, and the D part goes and binds to the SRE, the steroid recognition receptor element, in the DNA. At this point, when this D binds to the SRE, it initiates transcription. It initiates transcription. So any gene that has an SRE sequence in front of it will be transcribed. Well, one of these happens to be HMG-CoA reductase. So when cholesterol levels fall, the cell starts to synthesize HMG-CoA reductase. Only when the levels fall does this start to happen. If the cells have plenty of cholesterol, they don't go through this process. They don't make that HMG-CoA reductase. Does that make sense? Yeah, so there's a lot of SRE things there, aren't there? Okay? So the SREBP is something that's binding to the SRE. And specifically, it's the D part of it that's doing that. Two hands in front, yes. Okay, can I put my cursor over the M part? The M part is the membrane part. That's this thing here. Okay? The membrane part has two parts. We're not going to take it any further than that. We're not going to call it M1 and M2. Okay? Let me get over here first. How is the SREBP and the ER regenerated? It's remade. So it's a good question. His question is, how is this regenerated? It's not regenerated. It has to be remade. Yep, one-time use. Yes, sir.
The question is, how exactly, you could explain briefly, what exactly happens? So cholesterol drops. How does these get to know that? So then the reaction of transfer into the...
How? Okay, so his question is, how does the cell know when the cholesterol levels are falling to do all this? Well, this is actually built into the mechanism of the system. So the cholesterol falling causes, there's a sensor in the cell that causes this to be transported to the Golgi. So we've seen last term how proteins move from the endoplasmic reticulum to Golgi and so forth, and this relates to some sequences that are on them. Well, these sequences that are on here ultimately will target it for this, but they will only be moved if cholesterol levels fall.
I was just wondering, like maybe if there is enough big level of the cholesterol, somehow it inhibits this reaction to happen, or...
His question is, then how does high levels of cholesterol inhibit this from happening? Well, the movement is dependent upon the low cholesterol levels. So we don't need to, I can't go into the steps involved in this process, but suffice it to say that there are multiple steps that's involved in getting it from here down to here. And these will only occur when cholesterol levels fall. Yes? Does R dissociate from the membrane when it's been cut off? This will be targeted for destruction at some point. That's correct. Yes, back there.
Does SREBP-G or SREBP-ER include the orange part, the blue part, and the serine?
So the SREBP-ER and the SREBP-G includes only the blue. Only the blue. You see, it's the blue that's actually playing the important role here. This orange part over here is another membrane protein that's holding on to this guy. That's called SCAP. Okay, we're not going to worry about that here. Yes.
You said that when G binds the DNA, it would initiate transcription of anything with an SRE in front of it.
That's correct.
Is there anything else with SRE in front of it that we have to care about?
Okay, so his question is, are there other things, other genes that have SRE in front of them? It turns out that there are many genes that have SRE in front of them, but none that we're concerned with yet in this class. Do they act in complementary ways? Yes, in fact, they do. They are all things that act in response to steroid binding. And steroid binding is actually quite interesting because steroids have an ability to cross membranes without proteins, it looks like. And we haven't talked about that, but the steroid hormones that are released can actually cross membranes on their own and will directly stimulate this process happening. But that's, again, beyond what we're going to talk about here.
Is it because they're lipid-based?
Because what?
Because they're lipid-based?
Is it because they're lipid-based? Probably the reason they can make it through is that they are fairly they're very hydrophobic. But exactly how they make it through, nobody has really worked out very well. Yeah. Question here. So the serine protease cuts this guy here. It cuts the R part off of the MD part. Yes. Okay? Yes?
So, if the D binds to the DNA, and that initiates the transcription of the SREB-G, how does that stop?
Okay, so he asks another good question. How is it that once we get this started, that ultimately this causes it to stop? And we'll talk about transcription later in the term, so I'm not going to address that here. But stopping transcription is a very important consideration. And you could imagine that binding of something like cholesterol might inhibit the binding of the protein to the DNA, and that, in fact, in many cases, is the way that that's done. Okay? Good question. Okay. Let's see. Anything I need to mop up about that? That I'm not going to talk about, and that I'm not going to talk about. Okay. So that is how the synthesis of HMG-CoA reductase is regulated. So we see that there is a lot of complexity to HMG-CoA reductase. One of the things I skipped over here was the degradation of HMG-CoA reductase. That's another way of stopping, okay, that protein from functioning and having to be controlled. All right? So HMG-CoA reductase is a very interesting enzyme, very, very tight, I shouldn't say tightly, but regulated in many, many different ways. And it probably is regulated in so many different ways because the cell really has to be able to respond to some very different needs for cholesterol, depending upon the circumstances it finds itself in. Okay, that's the place where we will stop the material for exam number 1. What I want to do now, and the number of questions that you're asking me here are very good. You'll probably have even more as I talk about the next part, which is the movement of cholesterol in the body. All right? This one always generates a lot of questions about the movement, and there are things about the movement that we don't know, as you're going to see. Okay? Many things about the movement that we don't know. Well, movement of lipids, and this is lipids as a category, not just cholesterol, but the movement of lipids, things like fats, okay, fat-soluble vitamins, cholesterol, all these are lipids. These have a real problem to be moved in the body. And the reason is that they're water insoluble. And the stuff that's flowing through our bloodstream is aqueous-based, water-based. Okay? So cells can't just dump out a bunch of cholesterol or a bunch of fat or whatever into the bloodstream and expect it's going to just go, because it won't. Water and oil don't mix. Water and lipids don't mix. So cells have to package these things up to travel in that aqueous environment. Okay? When I say package things up, I'm talking specifically about using things called lipoprotein complexes. All right? There are several lipoprotein complexes that are necessary for the movement of lipids in the body. Okay? You can see on this table there's one called chylomicrons, very low density lipoprotein, and these are all complexes. Intermediate density lipoprotein complexes, low density lipoprotein complexes, and high density lipoprotein complexes. You may have heard of these over the years be referred to as VLDLs in the case of very low density lipoprotein complexes, IDLs as the intermediate density lipoprotein complexes, LDLs for the low densities, and HDLs for the high densities. Yes?
What is the column apolipoprotein?
I'm sorry?
There's a column
Okay.
apolipoprotein.
So his question is, what does this column here refer to? This refers to the proteins that are found in these complexes. Okay? And these are lipoprotein complexes. So a lipoprotein complex has lipid and it has protein. The lipid is there to help hold the lipids that are put into it. The proteins are there to help to solubilize everything that's there. Okay? Okay. Good question. Now, the thing I want to show you about these is let's look at their density and let's look at their size. We're not going to memorize these numbers. Don't worry about memorizing numbers. But look at these numbers. What's the biggest one there? The biggest one there, the chylomicrons. Okay? Look at their size, 7,500 to 1,200 nanometers in size. What's the densest ones there? The densest ones there are the high-density lipoprotein complexes. The lowest density are the chylomicrons. This means that chylomicrons are great, big, fluffy complexes. Great, big, fluffy complexes. Now, I want to tell you a little bit about how each of these works, all right? And then tell you something that may surprise you. Okay? So let's look to see how these guys work. All right? That's not what I wanted. I thought I had. Here we go. Here's what I'm after. Okay. So this schematically shows the movement of all of these complexes in the body. All right? It shows the movement of all of these complexes in the body. We start with dietary lipids. All right? We'll start with dietary lipids. If I go to McDonald's and I eat a giant double cheeseburger with a big heaping thing of fries on the side, I just got myself a whole bunch of lipids. Specifically, I have a whole bunch of fat that was in there. I may have gotten some tiny amount of vitamins that was in there, and I probably got a fair amount of cholesterol that was in there. Okay? All of those are water insoluble. So the body has to solubilize things that aren't soluble in water, and it does it, okay, starting in the intestine. Okay? In the intestines, we have things called bile acids. Bile acids I referred to in the last lecture, that they are compounds derived from cholesterol, and they act like a detergent. They have a portion of them that is very polar, and a portion of them that is very nonpolar. And we remember that's what detergents chemically have to have in order to solubilize grease and fat and other lipids. In the intestine, our body mixes these guys together and actually churns them up a little bit so that those detergents in the forms of the bile acids can solubilize all the lipids that are in our diet. Okay? Once it gets them solubilized, it passes them through the intestinal wall. Okay? And as they pass through the intestinal wall, they get packaged into the first set of complexes, known as the chylomicrons. This actually happens in the lymph system. Okay? This happens in the lymph system. The chylomicrons, at this point, are full of fat, fat-soluble vitamins, and cholesterol. All right? They travel from the lymph system into the bloodstream. Now, here's these great, big, honking, fluffy things full of goodies. Fat, fat-soluble vitamins, and cholesterol. When they get in the bloodstream, they start to pass wherever the blood goes, of course. And they get to the capillaries, which are little bitty, teeny, tiny things, and they get stuck because these are great, big, honking complexes. Okay? Now, you might think at this point, now I understand why heart attacks happen. And you would be exactly 100% wrong. Okay? Heart attacks happen for other reasons. All right? Where are your capillaries? Well, your capillaries are in tissues that are needing oxygen. They're needing nutrients. Muscles are a really good example. Okay? So what's going to happen there is that you're going to have there are going to be enzymes that are at those tissues that are going to start chewing on the contents of the chylomicrons. They're going to eat away the centers of those, and they're going to take the things that they want. They will take the fats. They will take the fat-soluble vitamins. They may break off some fatty acids and take them, but they're not going to touch the cholesterol. Okay? So I always use the analogy of the Oreo cookie with the center being eaten away, and the center of the Oreo is definitely being eaten away by these enzymes. Okay? These enzymes are called lipases, L-I-P-A-S-E-S. Lipases basically derives its name from the fact that they are breaking down fat. Yes, sir?
So those enzymes are in the cells that surround the capillaries where the chylomicrons are being stuck?
That's correct. So his question was, do these enzymes surround the cells in the place where these chylomicrons are stuck? And the answer is yes, they do. They're extracellular. Okay? Well, when that happens, if you take an Oreo cookie, especially if it's a double stuffed Oreo, and you start eating the center away, it's going to get smaller and smaller. And you're left behind with something called chylomicron remnants. The remnants will be smaller than the chylomicrons. They'll contain less fat. Excuse me. They'll contain less fat-soluble vitamins, but they'll contain the same amount of cholesterol. Okay? They shrink enough on this digestion that they pass through the capillaries. So now they're able to move through the capillaries, and they then go through the capillaries and they head straight for the liver. The liver has a receptor that grabs a hold of those and absorbs them, if it can. Well, it'll absorb them. I shouldn't say if it can. It will absorb them. But liver has a limited capacity to handle things. We'll see that actually contributes to high cholesterol problems. Okay? If you're having weight issues, you're overloading your liver, you're getting too much cholesterol, you're getting too many things that you shouldn't, your liver doesn't have an infinite capacity. We'll see that the liver has the ability to put these things back out into the bloodstream. They do get absorbed first, though. Okay? So, the remnants have gone, they've hit a receptor, they have gotten absorbed into the liver. Okay? Well, that's fine and dandy. As I said, we don't have an infinite capacity. The liver, just like it plays the role controlling how much glucose is in the bloodstream, plays a very important role in determining how much fat and cholesterol is in the bloodstream. Okay? Liver is a pretty interesting and important organ. All right. The liver is sitting here, and it has to make a decision. Does the body need more lipids, including cholesterol, or is the body happy? Okay? Well, how does it measure that? Right? Well, what it does is it has a receptor on its surface called an LDL receptor. LDLs are one of those complexes of proteins that I was talking about, lipoprotein complexes. All right? It was, okay, the next to the most dense. Only the high-density ones were more dense. There are receptors for the LDLs that if the liver is binding a lot of LDLs, it says, oh, the body isn't using all the things I'm putting out. I'm not going to put any more out. Because it's putting out things that will become LDLs. So the liver, when it senses that the body needs some, will send out some complexes, and when they start coming back as LDLs, it says it doesn't need any more. Does that make sense? So the liver uses the level of LDLs to tell itself how much cholesterol, how much fat, and so forth does the body need. Okay? Well, what happens when it decides it's running out? The body needs some stuff. Well, here's what the liver does. It says, okay, I've got all this fat, all this cholesterol, all these fat-soluble vitamins. By the way, vitamin A is stored in the liver. Okay? If you ever want a high source of vitamin A, that's where it's kept in the body. Polar bear liver will kill you if you eat it because it's so rich in vitamin A that vitamin A is toxic in high levels. All right. High levels, so it's got these things sitting around itself. All right? How many people have ever eaten polar bear liver? It doesn't happen too often, does it? You know, just a little bit. Okay. The liver senses the body needs some stuff. What does it do? It packages up a bunch of lipids and puts it into complexes called the very low density lipoprotein complexes, VLDLs. The VLDLs go out and they have the same fate as the chylomicrons. They go out, they get stuck, and they get their insides chewed away. As they start losing things, their density changes. They become intermediate density lipoprotein complexes. And again, the things that are being chewed away are not cholesterol. The intermediate density lipoprotein complexes lose some of their goodies and become low density lipoprotein complexes. And that's the LDLs. Now, if the cell is needing cholesterol, what it will do is it has a receptor on its surface that we've talked about previously that will bind to the LDL. It's called an LDL receptor. And it will absorb, it will internalize that LDL, taking all the cholesterol inside with it. Okay? Now we begin to see if cells aren't taking in LDLs, what would cause the what would happen to LDL concentrations? It would go up. As it goes up, the liver says up. It's not taking up LDLs. Don't need any more cholesterol. Right? On the other hand, if the cells are taking up LDLs and the LDL concentration remains low, the liver knows, whoa, it's taken out everything I'm giving it. I should give it some more. Right? Very, very important thing. Now, that's if everything's working perfectly. We know, of course, that it doesn't always work perfectly because we know many people who have high cholesterol. Why will people have high cholesterol? There's many causes of high cholesterol. We probably don't know all of them. We know diet is a factor. The more cholesterol you take in, the more dependent you are on this system working and functioning properly, and the more dependent you are on the capacity of your liver. If the liver gets full of stuff, it's got no choice, folks, but to start dumping stuff out into the body. So one of the first things that your doctor is going to do if the doctor discovers you have high cholesterol is not going to give you drugs. The doctor is going to try to control your cholesterol with diet. Can you change your diet? Can you change what you're eating so we're not overloading the liver? And see, do the cholesterol levels fall? That's the ideal world. You don't have to take drugs. Your cholesterol levels fall. If the person is either not able to maintain the diet or the diet isn't doing any good and the cholesterol levels remain high, then they will move to treating it with statins that I talked about last time. Yes?
If you have high cholesterol and the cells are not taking it on to stay in the bloodstream, you will actually be slowed down.
I'm not sure I understand the question.
Well, I feel like the problem with having high cholesterol would be that your body's taking it, like the tissues are taking it, but if the tissues don't take it in, you want it to stay.
Okay, so his question is the problem with high cholesterol the fact that the tissues are uptaking it? No. The problem with high cholesterol is the fact that your blood cholesterol levels are high because they're not uptaking it. So LDLs are also referred to by many people as bad cholesterol because it's the LDLs that are associated with atherosclerosis, which is associated with heart attacks. All right? LDLs are the bad cholesterol. They are the ones associated with it. So the more LDLs you have floating around in your bloodstream, the more likely you are to have an atherosclerotic plaque, and this is literally a plug that will occur in your arteries, stop blood flow and cause a heart attack or cause a stroke. Why do LDLs do that? Okay. LDLs do that because LDLs are more prone to oxidation. Oxidation happens from reactive oxygen species. Reactive oxygen species come from, smoking, things like that. Smokers have a much higher tendency to have atherosclerosis than non-smokers do. Okay? The oxidation of the LDLs causes them to be recognized by the immune system, and the immune system attacks the oxidized LDLs. Macrophages will attack the oxidized LDLs. And in attacking them, did it shut up? It did shut up. Nope. Battery is acting up. There we are. Okay. In attacking them, all right, they can themselves all become attached to the arterial wall. So this complex of LDLs that are oxidized, macrophages, these things grow. It gets rather ugly. It forms something we call foam cells because it's so foamy looking, contributed to that partly by the fact that cholesterol is there. Over time, this foam cell complex can become a block, a physical block. And that's what leads to atherosclerosis. That's what leads to the heart attack, stroke problem associated with these. All right? This thing is acting up. we interrupt this program for a battery change. Yes?
Is that why if you take hormones, your cholesterol levels go up? It's not being used to produce those hormones?
What hormones are we talking about?
Like if you have to take hormones,
Yeah, to be honest with you, I can't answer the question. I don't know. His question was, if you're taking hormones, sex hormones, or something like that, well, is that why your cholesterol will go up? Perhaps, but I just don't. I don't know the answer to the question. Yeah, sorry. You can sing, but we're not going to call it a day. How's that? Okay, now I think we're back on. Okay, batteries give out. All right, now, there's a lot of complexity here. Now, let me show you a couple things. I mentioned to you folks earlier in the term about the student I had whose son had the familial hypercholesterolemia. Did I mention that to you? I did, didn't I? No? Oh, okay. All right, then I got a story for you. Better than a song. Look at this. I had a student. I taught this class about 10 years ago, and a student came up to me after class and said, my son has a disease you've been talking about. Well, the disease I was talking about is called familial hypercholesterolemia. All right? It arises because the people who have this genetic disease are unable to make LDL receptors on their liver. They can't make LDL receptors on their liver. Well, what role did the LDL receptor play in that scheme I just showed you? It told the liver that there's enough LDLs or there's not enough LDLs, right? Well, imagine the liver has no LDL receptors. The liver is going to always think there's not enough LDLs. So what's the liver going to do? It's going to package up and send out as much VLDLs and LDLs and all the various complexes as it can to try to feed the body. And it continually comes back with, well, that wasn't enough. That wasn't enough. Okay? All right. Anyway, I'll get to this picture in a second. Okay? And by the way, she gave me this picture with permission, so I'm not just using this picture without permission. This child, okay, was about 5 years old when they discovered that he had these things that were arising on his knees and his elbows. And parents said, what the heck is this? Don't know what this is. Okay? And take him into the doctor, and the doctor doesn't know what it is because this disease that this kid has is so rare that hardly anybody ever sees it. The reason they hardly ever see it is most people die of it. Okay? You'll see why in a second. All right? These were basically packets of cholesterol. Okay? Now, to give you an idea, a person who has high cholesterol, doctors start getting worried with a cholesterol reading of about 200. They'll start giving people drugs when they get to that point. This young man had a cholesterol reading at 5 years old of 700. 700. Most people who have this disease are dead of heart attack by age 10. They found him at age 5. Okay? It's extraordinarily rare. Okay? There's maybe 10 people in the country that might have it. When she told me this, I said, there's no way. No, she said, I'm pretty sure that's what he's got. Okay? Well, sure enough, he did. Okay? Fortunately for him, statins had just been discovered, and been, I shouldn't say just been, but had recently been used for treatment and so forth, and some other cholesterol-fighting drugs came out to where they actually were able to treat him and got his cholesterol level down into the about 200 level, which is still fairly high, but probably which would lead him to live a reasonably normal life. Okay? Really a bizarre story. So I guess I've learned from that you should always listen to your students when they say they know something. So, and she was right. So, interesting story. Okay, so the consequences of having, all right, no receptors are really problematic. Well, I've given you the bad news. The good news is that we have something in our body we call good cholesterol. Good cholesterol is also referred to as high-density lipoprotein complexes, HDLs. HDLs are sort of assembled from some of the remnants of various things floating around, package it up with some other proteins, and HDLs actually help to lower cholesterol problems in the bloodstream. It can actually grab cholesterol that has gotten loose. It can grab some of the complexes that are not functioning properly and haul everybody back to the liver, where the liver can reabsorb them and try to get things right. Okay? If you have high cholesterol, one of the first questions that your doctor is going to have is, what's your level of HDL? Because high levels of HDL are considered good, and they're associated with the opposite of high levels of LDLs. Okay? How do you increase your HDLs? Stop smoking. Exercise. Okay? Watch what you eat. Although every time I say that, I get some various people saying, that's not quite true. Okay, well, be careful what you eat. All right? All right? Those things are associated with increasing levels of HDLs. HDLs do play a very good preventative role in stopping some of these problems from happening, such as we associate with atherosclerosis. Okay, questions? I may have a few at that point. So questions about what I've been talking about? Yes? So what does it mean to say you have genetically high cholesterol? Okay, it can mean a variety of things. All right? It can mean a variety of things. It can mean that perhaps your liver isn't processing things the way that it should be. It could mean that your receptor is perhaps slightly mutated but partially functional. Okay? It could mean that your diet, the way that you're handling things up here, are getting dumped into the bloodstream in different ways that are being absorbed differently. So there's many, many factors that may play into genetically predisposed high levels of cholesterol. Yes, sir?
What's the middle section there with the SFAs? What is that?
Okay. What's this part right here? It's not the future farmers of America, I can tell you that. Okay? Chylomicrons do not turn into future farmers of America. Okay? One of the things that happened was I said these guys can go out to the cells and enzymes can attack them. Thank you for asking the question, by the way, because this clears up a point. The cells can attack them. Enzymes can start eating away the center. Many of the things will get internalized, but some of those things will get away. Some of the things that get away are free fatty acids. So when you break down fat, you produce free fatty acids, and they can escape. Okay? Free fatty acids in the bloodstream cause a problem. Anybody know why free fatty acids all by themselves in the bloodstream would cause a problem? They aggregate together. Excellent, right? And what does aggregating together happen? What does it do? They blob up, and they act like a detergent. That's what detergents are, right? What do detergents do to your proteins? Denature, right? So we don't want to have free fatty acids floating around in our bloodstream, so we have a protein that mops them up really well. It's one of the most abundant proteins in your bloodstream. Anybody know what it is? It's called serum albumin. Serum albumin grabs free fatty acids and carries them so that they don't cause other problems. Yes? How do very low density lipoproteins get released? They're actually made by the liver. What signals the liver to release very low density lipoprotein complexes? I'll let somebody else answer that. What's going to be the signal to the liver to release more VLDLs? LDLs, okay, aren't binding to the LDL receptor. Because the liver is using the level of LDLs binding to its receptor to indicate how many of these guys are getting taken up. So if there's no LDLs being bound by the receptor or low levels of it being bound by the liver receptor, that means the body is taking the ones out and it needs to release more. Okay? This is also why you hear these stories about people who can eat a dozen eggs for breakfast every day and they have perfectly normal levels of cholesterol. What does this tell us? It tells us that this system is working well in them, the way it's supposed to. Yes?
So you said the LDL, they're susceptible to oxidation, right?
Yes.
And then the macrophages come and attack them, and they're the ones that cause the atherosclerosis.
Right.
So people who are on immunosuppressants like prednisone, would that lower atherosclerosis?
So prednisone meaning...
Like an
so,
immunosuppressant steroid, if you were on that.
Oh, I see what you're saying. Okay. So that's a good question. So his question is, since the immune system is involved in attacking the damaged LDLs, if you're taking an immune suppressant like prednisone or something like that, will you have lower levels of atherosclerosis? My suspicion is you probably would, but again, I'm not a doctor, so I try to be careful with that. So, prednisone has other effects, so you wouldn't take it routinely. Yes?
So the LDL that binds to receptors must be intact, right? That those ones who were taken by body and pretty much torn apart by enzymes in the cells and capillaries, next to capillaries.
What's being torn apart? I didn't understand.
So we say like, okay, LDL receptors, right? So it must be intact LDL that bind to the receptor.
Oh, okay. Oh, I see. Okay, so his question is, does the LDL have to be intact in order for the LDL receptor to bind it? That's your question. It turns out that there's a specific protein within the LDL that is being bound by the receptor, so it doesn't have to be completely intact. No. Yeah. Yes?
What happens when they reach the end of that arrow going to the liver? What does the liver do?
Oh, okay, I'm sorry. I didn't understand your question. I didn't finish the picture, so I should. Free fatty acids, okay, can be absorbed by the tissues. All right? Tissues can use them for energy or make fat. The free fatty acids can also be stored as fat in adipose tissue, which is what fat cells are. Okay? Fat cells can release free fatty acids as needed by the body, just like the liver can release fat and so forth as needed by the body. And those free fatty acids can ultimately make their way back into the liver. That's really all the rest of that's showing.
And then it just doesn't do anything that can be used again.
Yeah, so the liver is just like a big, bank to hold all this stuff is really what it is. That's right. That's right.
What about, so we say free fatty acids that go to cells, and it seems like then cells eject the excess of it in terms of the fact in HDL and send it back to liver. I'm talking about this red arrow that goes right after the cell picture.
Right here.
No, red cell. Or red
Here?
Red arrow.
Okay, this arrow.
Yes.
Okay, so this is the tissues are packaging, as I said, things are repackaged to make HDLs, and the HDLs are coming back over here. Okay? I hear the rustling. We're not quite done. Any other questions? Yes? So does the binding of the HDL by the HDL receptor in the liver have any effect? That's a very good question also. It doesn't have the same, as pronounced of an effect as LDL does, but I think it does, in fact, affect the liver's output of VLDLs. Yes, it would. Okay? Other questions? Okay, so two things. One, remember when you come in next time how we position ourselves in here? So every other seat, the first 10 rows sit everywhere. After that, then it's every other seat. 1, 3, 5. If somebody is sitting in 2, don't sit in 4. All right? So 1, 3, 5, 1, 3, 5, 1, 3, 5. That's top and bottom. Okay? And the last thing is, I think we should sing a song. Oop. Hold on. Start over. All right.